Hi, everybody. This is going to be a relatively brief recording of just a quick review for whenever you take the midterm, which should be coming maybe week 5, and you probably should be taking it around week 7. So you're not going to see a whole lot from a screen perspective, but you'll hear my voice. That's there. So it only consists of 40 questions. It's 20 apiece from Dr. Buxton and from myself. So here we go. We'll take a quick look see and see what's going on with it. I do like to function a lot on the cell cycle because early on, that's really what we're talking about is the impact. First, understanding how cells work, and then the impact of radiation upon them. So, what ionizing radiation? Remember the distinction. Ionizing radiation is what you're going to be utilizing more so, particularly with X-rays or CT scans or radiation therapy or that type of intervention that's going on with nuclear medicine and or the exposure to various nucleotide labeled substances that or substances that are really by definition utilized in imaging where basically they're labeled in such a way that they can be ascertained their presence in various locations by what they're attached to, typically things like technetium, even though it's a comparatively minor amount of radiation exposure. But what happens in cells, okay, is they develop mutations from ionizing radiation. It actually causes DNA strands to break, most likely through a combination of toxic derivatives of oxygen and exposure time and the fragility of the cells and nutrition and a variety of factors that you've been learning about throughout the term. Well, the first thing, what's a mutation? An error in DNA. So, where is DNA assembled in the cell cycle? And you can go back and look at that. It looks like just a big circle, the cell cycle, and there's different phases. There's the standard mitotic phases, prophase, metaphase, telophase, anaphase, and cytokinesis is the actual cell division. Remember, mitosis is nuclear division, but it's what happens before that. And that's G1, S, G2. And if it exits that cycle and is no longer actively reproducing, we say it's in G0. So mutations occur when DNA is assembled. When's DNA assembled? S phase. And so when there's a couple of checkpoints, and what will impact those checkpoints is an accumulation of certain products. If the checkpoints are not met, the cell won't replicate. And sometimes, if there's a sufficient amount of accumulation of problems, the cell will literally, if you want to put it in this fashion, commit suicide by opening up little packets of enzymes called lysozymes. Lysosomes are the packets, lysozymes are the enzymes, and those will basically cause the cell to die. And we call it programmed cell death, or it consumes itself. Sometimes it's called autophagy for self eating, but really most of us call it apoptosis, programmed cell death. So these are the important areas, and maybe the DNA is the most important part where mutations occur. Though usually when we see the apoptosis from that, it's in G2. But again, we're DNA synthesized. It's not so much where DNA is synthesized as much as when it's assembled, and that's really what S phase is about. The building blocks are probably put together in G1. But the assembly and the paired chains, the complementary pairing and the duplicating of the chains, that's really where we see it. So that's really, if you're talking about replication of DNA, that's the strand being assembled. And the goal of the exercise is certainly to produce mitotically equivalent cells. The whole idea of mitosis, which is really how our body repairs itself. It doesn't repair itself. To be truthful, it replaces itself. We get rid of what doesn't work, and we build new ones if we can. New ones that should function just like the original model, so to speak. And that comes from mitosis. And mitosis is producing daughter cells that are genetically identical. Its opposite is meiosis. That's sexual reproduction. And you and I, that only occurs in eggs and sperm when they're produced in the ovaries and the testes. Okay. And that's to produce one basically a limited number of so quote unquote daughter cells that are not genetically identical but genetically diverse, and indeed they only hold half the genetic information. So there's a whole different structure. So when meiosis is actually accomplished and then eventually the egg and the sperm fuse, you really get what most of the time are two genetically distinct components from the mom and the dad. We end up with a fusion. Fancy name is called a zygote, the fertilized egg, and then as it replicates by mitosis, you really have something that's distinct. That should be most time. I mean, there's going to be similarities, but distinctly different. What happens when, and this is really where our focal point is, both of what we could conceivably cause, but most likely what we treat in intervention with radiation therapy are cancer cells. And cancer cells, when the cell cycle has lost, because really it's a controlling cycle. Remember what the cell cycle is. The cell cycle is sort of like the rate and governing replication. And in our bodies, we govern, we hold the cycle. The cycle will go too fast. So it's almost like we're kind of like going down a hill and we're putting brakes on it. And if we lose the brakes, that's really a big problem. That's the cell cycle. So it loses control. It doesn't undergo apoptosis. Mutations occur. Cells grow in excessive amounts. They grow irregularly. The cells are not typically not normal cells. They're aberrant cells. They're invasive. They just don't even behave like the cells are supposed to be replacing. That's really the problem. Interestingly enough, certain cells that surprise you that are already formed are more susceptible to radiation than others. Cells, well, if it doesn't have a nucleus like a red blood cell, wouldn't be sensitive. Nerve and muscle cells, whether it's heart muscle, which is cardiac muscle, or smooth or skeletal muscle, or nerve cells, basically are not. Uh, White blood cells are surprisingly susceptible with regard to those things. And what's interesting is when radiation occurs, we really begin to see when a cell is exposed, once the DNA is assembled in the cell cycle and it moves on to G sub 2, which is sort of like preparation for division, and M phase, which is actual division. That's typically when the cells are most susceptible to damage. So, in general, we have like a laundry list of cells that are, and it's all based on how rapidly they reproduce, is how susceptible they are to damage from radiation. And the ones that we reproduce the most, by far, the most rapidly, not necessarily the most in number, but the most rapidly, are lining cells. And I would think, to be fair, the most in number as well. For instance, we probably make a billion gastrointestinal lining cells every day. We replace them all every 3 to 6 days. In the adult, that's probably the fastest replicating cell. When we look at the embryonic and fetal tissue, a little bit different because those cells are just growing super rapidly as the baby develops before it's born. Then connective tissue, then things like muscle and nerve tissue. That's sort of the laundry list of when these cells are most susceptible. The epithelial cells, because they replicate the fastest. The connective tissue cells, many of which are still replicating. Then muscle and nerve cells with regard to susceptibility. In particular, those gastrointestinal cells, the ones to watch out for, are particularly sensitive to radiation and particularly sensitive to damage, are located in the small intestine. And that's real, and they're their busiest because that's where the vast majority of digestion and absorption occurs. And the areas, there's little indentations where these cells are being reproduced, and they have a fancy name. They're called crypts, CRYPTS. In the stomach, you have pits, and in the small intestine, you have crypts. They have a fancy last name called crypts of Lieberkühn, but nobody really cares about the fancy name that's associated with it. And so that's really where the ones that are most commonly affected. And typically, what you see with exposure to radiation with most individuals is diarrhea, gastrointestinal problems. Those are typically the first ones that show up. I can't even tell you how many patients I've had over the years. Not that I was necessarily prescribing the treatment, but I treated mostly older folks who had, and I was in an area that had a very high cancer rate. When I practiced, it was big industrial, basic industries, a lot of oil, a lot of steel, a lot of chemical plants, and boy, there's a lot of toxins that are in the air. and that people came in contact with. So it was environmentally not great. And people would come, oh yeah, I used to go home, tell my wife, my first wife at the time, and go, I swear to you, everybody in my office today has had one point either had or has cancer. It's crazy. Certain tissues that are well differentiated, and I commented on that in the recorded lectures. That means they've been assembled and they don't reproduce, are particularly fortunate not to be as susceptible to radiation. The other thing that really helps is not the bone marrow, but the external part of the bone because it's all calcium. And that calcium will help to absorb the radiation. It's like kind of an x ray. The bones show white. That's because an x ray is really kind of like it's similar to a negative from a film in the old days. And it's kind of like it kind of is almost like you're looking at the negative. So it hasn't been exposed. The film isn't exposed and turned black because it looks white because the radiation didn't get through the calcium. Is kind of what happened there. And don't forget about, I'm sure Dr. Buxton commented on things like shouldered curve. And that's what the shoulder curve is. It means that the damage is delayed. When we look over time, time being the x-axis, the damage is relatively slight and it creates like a shoulder before, as it comes into sort of like the end of your shoulder as it going out to your arm, it kind of falls down. And it's called a shouldered curve. So the more it divides, initially it doesn't show up, but once it starts to divide more and more, then we begin to see problems associated with it. So it's called a shouldered curve. Okay. It's funny and it's sort of, and I love things that are counterintuitive. In cells that are damaged by radiation, things that actually help it repair are not having enough to eat, decreased areas of nutrition. It's really unusual about that. That's there. When you have all those nutrients, it allows the cells to divide. They become more, I think of it this way, they become more rapidly active when they really shouldn't be, when they almost should be sitting there. It's almost like they tell you, if you're sick, take some time and rest and take it easy rather than go back to your full activities. Kind of think, I don't think of it that way. That, in the early stages, particularly with damage exposure, cells that aren't as well nourished actually do better than not. And certain things that really sensitize you, because a lot of the effects of radiation are called toxic derivatives of oxygen. Anybody who utilizes oxygen makes toxic particles called free radicals. A lot of the things we eat, a lot of the things in our body gobble those up. We have special enzymes. When you hear about antioxidants foods and superfoods, they're rich in those enzymes. So they supplement us in fighting off these toxic derivatives of oxygen. So any organism, anything that lives that utilizes air, utilizes oxygen, makes these. So when you have a lot of that, interestingly enough, it tends to expose you more to problems. Okay. So it actually sensitizes you to radiation because there's more oxygen around. Whereas even certain proteins protect you because they absorb radiation better. There's a particular, it's called an amino acid. It's one, you probably learned about it. One of the building blocks of proteins, and why it has what's called sulfhydryl groups. It has sulfur, and sulfur sucks up the radiation a little bit better. That's there. And so its fancy name is called cysteine. Okay, there's cysteine and it changes shape and it's called homocysteine. It's not the same as cytosine, which is one of the building blocks for DNA: adenine, guanine, cytosine, and thymine. Cysteine, Cysteine, is how you spell it, that are there. So that's really, it's another one of those important ones. And the big player in that are these sulfhydryl groups that are there. Take a look, and you should review what goes on in M phase and mitosis in the cell cycle, some of the events that are there. So you'll be able to take a look at that. We don't start to see the DNA in interphase, which is G1S and G2. It doesn't even show up. It isn't until it goes into M or mitosis phase. At the start of it, something called prophase, the nuclear membrane disappears, the spindles, which is how the chromosomes are going to align, begins to show up. A thing called the nucleolus. There's a little nugget in there that appears to disappear. It just kind of loses its integrity, comes back. at the end of the cycle. But that's when we start to see the chromosomes. They actually start to supercoil and they become visible. That's one of the earliest signs that we see that a cell is getting ready to reproduce. And another problem associated with these things is water, because water inherently has a lot of oxygen as part of it. It's a component, and in the cytoplasm in the cell, where a lot of the damage is, about 90% water. So don't forget about that. So water and oxygen itself, oxygen, think of the oxygen gas, and the oxygen in the water are the big players, because a lot of times water will develop, the OH groups will develop free radicals as well. So it's the oxygen component in the water that's there. And when these free radicals make the DNA break, what happens is the DNA, if those breaks are really close together, it's very hard to assemble proteins, and that's really the worst damage. If you have let's say a strand that's just for argument's sake a foot long, and you have a break on one end near the end and a break on the other end near the end, nearly the whole is complete in the middle. So if you're just making the protein from the middle, it'll have no effect. But if you have breaks that are close to each other in the middle, you're going to probably interrupt something from assembling the protein. So breaks in both strands close to each other, big problems that are there. So, and the whole thing about DNA, look, we are, and I say it all the time, we are what our proteins say we are. And DNA is basically the owner's manual, it's the assembly instruction. Just like when you buy something and assembly is required, DNA is the manual that makes proteins, and we're made out of protein. So it's the assembly manual for making proteins. So don't forget that. And the other, that's the first 20 questions. The next 20 questions are from Dr. Buxton. He may or may not be providing you with some information about this separately. He usually does in certain ways if you go back and listen to what he's talking about. So please do that. And this concludes my part of the review. And I will incorporate this, and I'll send you a note that I'm incorporating this in the where it says midterm exam, so you can listen to it beforehand. Thank you. Bye bye.